previous lectures we have dealt with the basics in chromatography, that is we have discussed the basic concepts in chromatography, and we have also discussed about the various performance parameters of chromatography. Now, in this lecture we are going to extend our discussion on column chromatography, that is the most widely used chromatographic method. What are the basic components of column chromatography? Now, column chromatography consists of a column. A column could be made up of a glass, it could be a glass column or a metal column, or it could be a plastic column. Then another component is stationary phase, which is packed in the column. Now, it could be a simple stationary phase or a stationary phase supported on a matrix or support material. Then a mobile phase which passes through the stationary phase, and due to distribution coefficient the partitioning or the separation of analytes takes place. There is a pump or we can say delivery system for mobile phase, particularly in high pressure or high performance liquid chromatography techniques you certainly require a pump, and even in low pressure chromatography, column chromatography, you can do it by gravity feed, but it is better to have a pump like peristaltic pump for constant flow rate. Then you need a sample loading system. For low pressure chromatographic systems, you can load the samples manually also, but for advanced systems there has to be an injector system attached to the apparatus. Then finally, a detector and chart recorder. A detector, different types of detectors are used as we will see, and chart recorder gives the readout. Samples could be collected in a fraction collector if they are required for like say you want to purify or analyze them afterwards. So, in fraction collector different peaks can be collected and then it could be used for further analysis. Now, column chromatographic system can be classified according to the back pressure generated within the column during separation process. So, low pressure liquid chromatography systems generate pressure which is less than 5 bar or 0.1 megapascal. Medium pressure liquid chromatography generates pressure between 6 and 50 bar, and high pressure liquid chromatography generates pressure above 50 bar. Nowadays, like both medium pressure and high pressure gives excellent resolution, and this particular demarcation has been kind of gone, and now they are called, both of them are called high performance liquid chromatography system. So, HPLC or high performance is preferred rather than high pressure or medium pressure. All right. So, if you consider this is a very simple schematic of a chromatograph or equipment part. If you can see here, there is a column and there is a Marriott flask above this where the solvent or the mobile phase is there. Now, this will be run on gravity, and so although this kind of maintains the pressure, but still the flow rates are may vary as per the level of the mobile phase. Finally, this will go to detector, and as eluate comes out, it will go to fraction collector and through detector. The improved systems even for low pressure chromatography, here you can see one, there is a eluent reservoir and there is a pump, it is usually peristaltic pump, as you will see later on. The pump kind of derives the mobile phase into the column. There are two plungers here and this is nylon disc or nylon filters are attached to it. So, stationary phase is supported on to here. As the mobile phase passes, it comes out here and goes to detector, and finally, it could be collected in a fraction collector. So, that is a very simple arrangement, and we will discuss each of these things individually. So, let us start with the columns. Now, these columns, mostly these are glass columns are used, and should have, these columns should have a means of supporting the stationary phase near the base, and that is important. That first thing is you have to support, so that it could be staying there, and another is you have to minimize the dead space below the column support, so that post column mixing of separated analytes will not occur. Now, commercial columns possess either a porous glass plate fused on the base of the column, or a suitable device for supporting a replaceable nylon filter or net is there. So, which will support the stationary phase. So, a column, like I said, even syringe columns could be used, like syringes could be used as columns, and glass wool could be used to support the stationary phase for simple chromatography. Matrix materials which are used, which support the stationary phase or stationary phase is attached to them. Matrix materials should have, I will discuss about what kind of matrix materials are used, but they should have certain properties. It needs to have, say, high mechanical stability. To maintain good flow rates, you have to have the particles or the stationary phase with good mechanical stability. So, they are not compressed or flow rates are not altered actually. It should have good chemical stability, as you are going to use different kinds of buffers, solvents and other things. So, chemical stability is also required. Then there should be functional groups on stationary on matrix to facilitate the attachment of stationary phase, because most of the time, like say for attaching ionic groups or say affinity ligands, you need to have certain functional groups where these could be conveniently attached. Then it should have high capacity, that is you can support large stationary phase, so that amount of sample to be purified could be enhanced. Then they should be available stationary, this matrix should be available in a range of particle size and also in porous form for certain kinds of chromatographic methods. The surface of matrix needs to be inert to minimize non-specific adsorption of analytes, otherwise the chromatographic separation will suffer, as peak trailing or other problems might occur due to non-selective adsorption on the matrix. Now, there are different types of matrices which are being used, and some commonly used matrices are example agarose. Now, this agarose is a polysaccharide made up of D-galactose and 3,6-N-hydro-1-galactose units. It is the unbranched polysaccharide chains are cross-linked with agents such as 2,3-di-bromopropanol and to give gels that are stable in pH range from 3 to 14, and examples are like saffrose and biogel A, which are frequently used. Second material could be polyacrylamide. It is a polymer of acrylamide cross linked with bisacrylamide. You will see this is quite used, both agarose and polyacrylamide are used in electrophoresis in the next section. Polyacrylamide is also pretty stable in pH range of 2 to 11, and example is biogel P, that is a commercially available. Then cellulose could be used as matrix material. It is a polysaccharide of beta 1,4 linked glucose units and cross linked with epichlorohydrin. It has good pH stability and very good flow properties and is hydrophilic in nature. Dextran, that could be another polymer. It is a polysaccharide containing alpha 1,6 linked glucose units, and this will also be cross linked with epichlorohydrin. It is less stable to acid hydrolysis, and it is stable up to pH say 12. It is hydrophilic, and one example is Cefadex, which is commercially available in different forms. Then polystyrene, that is synthetic sort of material could be utilized. It is a polymer of styrene cross linked with divinyl benzene. It has a good stability over all pH and mostly used for exclusion and ion exchange chromatography. Then there is another one called silica. It is a polymeric material produced from orthosiliceous acid, and it is stable in pH range of 3 to 8. So, these are some of the examples of the matrices which are being used. Now, on these matrices attached is stationary phase which is involved in the purification process. So, the chemical nature of the stationary phase depends upon the particular form of chromatography to be carried out. Say if it is ion exchange, you will be immobilizing ionic groups, if it is affinity and so on. Now, most stationary phases are available attached to the matrices in a range of size and shape commercially. So, both properties of size and shape are important because they influence the flow rate and the resolution characteristics. Now, column packing, once you have selected the matrix with stationary phase, the next step is to pack the column with the stationary phase, and that is very important part of chromatographic separation. Now, packing a column is normally carried out by gently pouring a slurry of the stationary phase in the mobile phase into the column, and column is gently tapped to ensure that no air bubbles are trapped and the packing settles evenly. Now, poor column packing will give rise to uneven flow and reduced resolution, and once a column has been prepared, it is very important that it does not to make sure that it does not run dry. So, it should be all the time kept wet actually, or mobile phase should be there. If it gets dried, then it will break or crack, and that again you have to maybe pack it again. So, let me show you how column is packed and how sample is applied onto the column on your screen. So, please see on your screen. So, what is done, if you have a glass column or any other column, and you can see there is a support at the base which will stabilize the stationary phase. Now, what happens is your stationary phase is filled in through a glass rod. Either you have to have a glass rod here, and you will pour your stationary phase onto the through this glass rod, so that it will pass and it will settle down, it will go to the base through the walls of the column. Now, advantage is that you will and it will be filled from the base, that if you suddenly add this to the column, the air bubbles will be trapped, but with this method air bubbles will not be trapped at the base of the column. Other way could be that you can tilt the column, rather than if you do not want to use the glass rod, then column can be tilted and then material could be poured in through the sides of the wall. So, this way what you can do is you can pour and see that when you are pouring this material, you do it evenly, like continuously, without without creating gaps or demarcations. Like for example, there could be a column which could be packed in this way, that there is a some unfilled areas could be there, and this will again give uneven flows and will hamper the chromatographic procedure separation. So, these things has to be looked into. Now, how sample is applied onto the column, let us see that. So, when you have to apply the sample onto the column, what is done is, again you have a packed column. Once you have packed and you have made sure you have equilibrated the column with the particular buffer in which chromatographic separation needs to be done, then you have equilibrated the column or stationary phase with that. Now, so what you will have is, you have stationary phase filled in here, and you have your mobile phase which is here, and you will have either you will have closed it with plunger or with a certain cork with tubing in that. So, there will be tubing which is attached to your mobile phase. It may be through pump or it may be without pumps. So, you can do it either by gravity or maybe without gravity. Now, how do you load your sample? One is, if you have lot of sample and it is like say ion exchange chromatography things, it could be directly loaded through peristaltic pump. But other methods could be that you have to remove this top plunger or cork whichever is there, and then what you have to do is, then once you have this material, and so you have to remove this mobile phase from here very slowly without disturbing the stationary phase bed. Once you have removed, then very nicely with a micro syringe or a pipette uh, you can inject the sample, or you can load the sample in here in a smaller volume, and then very slowly let this sample get into the column, and once it gets into the stationary phase, then you can put the mobile phase again, and then put the plunger on and start the chromatographic procedure. Another method could be, you could have high density solutions, like you can use sucrose density, sucrose or glycerol in your sample, which gives them high density, and then with you do not have to really remove this mobile phase, because you can put in when your mobile phase is there, you can put in the material, and since it has high density, it will settle in here without any problem, and so you will be able to do it without removing the mobile phase. So, these are a few ways you can do it, like say for high concentration of protein solutions are quite dense, and they could be done like that. All right, let us come back to our discussion. And so, this was about the column packing, and remember, poor column packing will certainly rise to uneven flow and will certainly reduce the resolution. So, it is very important that no air bubbles, no gaps, no uneven settling of the stationary phase, and all these things has to be taken care of. Then application of sample, as we have discussed, is a very important part that you can do manually or through injector system. Mostly in low pressure chromatography you can do it manually, like I have shown you, like you can remove the mobile phase, or you can use a high density like sucrose or glycerol, or you can use peristaltic pump. So, once the sample is loaded, then you have to develop the column and elute the analyte. So, the components of the applied samples are separated by continuous passage of the mobile phase through the column. Now, the separated analytes will be removed from the column by elution, and this elution process is essentially that the flow of mobile phase is maintained at a constant rate, and in certain systems the column certain systems, the analyte as per retention time will stay in the column and then slowly will be moved, and since they have different distribution coefficient, they will be separated and they will be retained separately or differently, they will be removed. So, it is a isocratic elution. There could be also gradient elution, where you are utilizing an eluent containing other substances, like say for example, salt elution. You might use sodium chloride, and that replaces the analyte, and then analyte is eluted. So, there you can have gradient elution, or that could be step gradient, or that could be continuous gradient elution could be done, and there are gradient makers which are available to make the elution to make the gradient. To produce a suitable gradient, 2 or more eluents have to be mixed. Like for example, you can have 0 molar NaCl in a buffer, and you can have say 1 molar NaCl in that buffer, and though 2 can be combined in correct proportion prior to entering the column. This could be achieved by using 2 different peristaltic pumps to deliver the separate eluents, and they will be mixed in a chamber before entering the column. Gradient could be a linear gradient, or it could be a convex or concave gradient. Convex gradients give better resolution initially, whereas concave will give better resolution at the end. This is a peristaltic pump, a very simple schematic of a peristaltic pump. It is a roller type peristaltic pump, and you can see these rollers here. What they do is they compress the tubing, and as they revolve, it's this revolution occurs. These rollers move, compressing the tubing, and due to the compression, the mobile phase is sucked in from the reservoir, and then will be at fixed flow rate, it will be supplied to the column. Now, this has to be standardized in a sense that different tubings will give you different flow rates as per the revolution of this disc actually. So, that has to be standardized, but that is a very simple peristaltic pumps. There is planetary type also are available, and both of them can be used for providing even flow or a constant flow. This figure shows this is a gradient maker. You have one solution here that is donor chamber, one is recipient chamber, and then as it could be mixed with a magnetic stirrer and a gradient could be made in a different proportion. This is like a linear gradient, or you can have convex or concave gradient as per your requirement. So, this is a very simple method of elution where you get your analyte out and you can collect it in a fraction collector, you can detect it through a different kinds of detectors. Method of detection and collection we will be discussing later on with HPLC section, as many things are very common. Only difference is detectors are for HPLC are more sensitive, and they are more quick to detect certain things rather than a low pressure chromatography system, where a simple UV monitor could be utilized. All right. So, this was about the low pressure chromatography system. Now, let us shift to high performance liquid chromatography and how you will see how it is different from low pressure chromatography. Now, the resolving power of chromatographic column increases with column length, as we were discussing earlier, and it depends on number of theoretical plates per unit length. But there is a limit to, or there is a practical limit to the length of the column because of peak broadening and other problems. So, what you have is that you cannot really increase the length to after certain particular length of the column. So, you cannot increase it to like there is a limit to increase of the column length. So, what has to be done to increase the theoretical plates? What could be done is that in a smaller or in a particular length column, you can have a stationary phase with support particles, of course, with a smaller particle size. So, if you have a smaller particle size of a stationary phase, the better resolution could be achieved, because you will have higher number of theoretical plates. As you can compare, there is a coarse material or higher size particles which will pack in the column, and they will certainly have low number of theoretical plates. But if you have very small particle size, they will pack much better and they will have higher number of theoretical plates. But problem is that as they pack very nicely, there will be lot of resistance due to smaller particle size, and which will create back pressure in the column, and simple pumps are not really sufficient to drive the flow, and many times this matrix collapses actually because of high pressure. Now, this problem has been solved by a lot of advancement, where development of small particle size stationary phase which can withstand these pressures. Very high pressures could be withstood by these particles, and so now this development has resulted in faster and much better resolution, and the HPLC or high performance liquid chromatography has emerged as the most popular, very powerful and versatile form of chromatography, and it is also very quick. It does not take much time as the low pressure chromatography, because it is done at very high flow rates or very high pressure limits. A very simple schematic of different components of isocratic HPLC system, if you see, what you have is a reservoir or eluent reservoir. Now, solvent reservoir, then there is a high pressure pump. So, we will discuss these are different from simple peristaltic pump, and then you have from pump, it is the solvent will be directed to the column. Many times there is a guard column here which kind of cleans up the system, and remember all mobile phase has to be degassed and filtered, so that it does not like precipitate things in there and leads to generation of high pressure because of that, because of the choking of the column. Column which is a small column, we will see how big these columns are. but length is certainly less than normal columns, because you can pack more number of theoretical plates in here. It goes to the detector and then to fraction collector. So you can collect it, you can detect, and readout could be seen. So, this is a very simple schematic of isocratic HPL system. If it is a gradient, then you have to have two pressure pumps and a gradient mixture, which will allow you to supply gradient and elute the sample. All right. So, let us discuss different things, different parts of this HPLC. First thing is, and most important, is column. Now, conventional column used for HPLC are generally made up of stainless steel, and they can withstand pressure, very high pressure, up to say 5.5 into 10 raised to power 7 pascals. Columns are generally 3 to 50 centimeter long and 4 millimeter in internal diameter, with flow rates which could reach up to 1 to 3 ml per minute. There are also micro bore or open tubular columns, and they have internal diameter of say 1 to 2 millimeter, and they are generally up to 50 centimeter long and can sustain flow rates of 5 to 20 microliter per minute. So, there are advantages also of micro bore columns, like the eluent consumption is reduced as due to slower flow rates, ideal for interfacing with mass spectrometer due to reduced flow rate, increased sensitivity and higher concentration of analyte could be used here. Now, matrices and stationary phase, if you see, there are three forms of stationary phase material are available. These are microporous supports, where micropores ramify through the particles, which are generally 5 to 10 millimeter in diameter. Then there is a pellicular support, which is porous particles coated onto the inert solid core, such as glass bead of about 40 millimeter in diameter, and then there is a bonded phase, where the stationary phase is chemically bonded onto the inert support, say silica. So, these are some of the stationary phases available. For adsorption chromatography, adsorbents such as silica or alumina are available as microporous or pellicular forms with a range of particle size. And for partition chromatographic system, the stationary phase may be coated onto the inert microporous or pellicular support. For ion exchange chromatographic system, cross-linked microporous polystyrene resins are widely used. Pellicular resins forms are also available, and a bonded phase exchanger covalently bonded to a cross-linked silicon network are also used. Now, application of sample is very important in this HPLC. If you could recall, in low pressure you can do it manually, but in HPLC, which is a advanced and enclosed system, there should be a very like very accurate or automated system of application of sample. So, application of sample onto HPLC columns is done through like for successful separation, it is done through a loop injectors actually and not manually. Now, this consists of a metal loop of fixed small volume, and they could be of varying volume from very small to very large, and that can be filled with the sample, and then eluent from the pump is then channeled through those loop by means of valve switching system, and the sample is flushed onto the column via the loop outlet without interrupting the flow of the eluent to the column. So, that is a very important part. Now, let us see, let me show you on your screen this loop injector, so that you can appreciate how does it look like and how this whole thing is happening. All right. So, loop injectors, like I said, they are very important part. Now, a loop injector is simply there are different ports, and let me make this for you here. It is a disc sort of, and you have different ports here, and we can number them like say 1, 2, 3, 4, 5, 6. Then there will be a loop which is connected. If you see, there is a loop here which is of So, this is a loop here which is being formed. Now, at this is a, now there is two positions, one is loading position. If I could say this is loading position, then what will happen? Loading position means you are loading your sample onto the loop. So, what is the configuration here? Configuration is that you have a particular eluent or the mobile phase from pump. This is going to the port number 1, and from the port number 2 it is going to the column. So, still you have not loaded. So, now this is the loading position where you can load your sample. If I say this is your knob which would has to be shifted, then this is the loading position, and this is your loop. This is loop is of fixed volume. Now, this could be, like I said, it could be like 200 microliter to say a few ml also. Now, what will happen? So, you have to load, this was the port where you will be loading the sample actually. So, this is inlet port for loading the loading sample by micro syringe. All right. Now, once you have loaded, so once you have put in your micro syringe and you are loading your sample through a micro syringe in this, you have to fix micro syringe. Then what will happen? This will go here, your sample will move from into this loop and it will fill this loop here. All right. So, what will happen? Say if this is 200 microliter loop. So, if you load 250 microliter, then 200 microliter will be contained in this loop, and rest of it will go to through the port number 5, like it is connected to 6, loop is connected to 6 and 6 is connected to 5 here. So, this will go to waste. So, these are both things will go to waste. So, what will happen? You have loaded the loop, extra sample will go to the waste. All right. Now, this is loading position. Now, let us see that you have to inject it onto the column. So, what will happen? What is the difference here? Now, what is, these are all things are same, that you have these ports here, which is 1, 2, now, and this is your loop here, and this is connected here. So, what was the situation here? Then here it is from eluent from pump is coming here, and this is to column. So, what will happen? Now, here if you see, this knob will shift to injecting position. Now, this knob which was here, this will be shifted to injecting position and it will be you can just rotate it to this position, and now the configuration will change. And the configuration will change in a sense that the eluent which was here going to port number 2 directly, rather as you change this knob, this will go to port number 6, and now port number 6 and 5 are not connected. And so, what is happening? The eluent is going to port number 6, then it goes to the loop, and from the loop finally it goes through port number 3 to port number 2 and then to the column. So, you can see the change here as you put it from loading to the injecting position, the eluent is directed through the loop, and so the eluent pushes the sample onto the column, and then this run could go on. When you have to inject again, what you will do? Again it will be brought to the loading position, and then again you have to do the same thing. These will go to the waste actually, both of them. So, this is the a very good system, injector loop which could be used for loading the or for application of the sample. All right. So, let us return to our discussion. All right. So, we were discussing about the application, and as I have shown you, this is how the application will be performed, application of sample will be performed. Now, mobile phase depends upon what separation needs to be achieved. If it is a isocratic elution, it could be made with a single pump using a single eluent or two or more eluents pre-mixed in a mixed proportion. But if it is a gradient elution, then one needs to use two pumps and there will be a gradient mixture or programmer has to be used. All eluents must be spatially purified because traces of impurities can affect the column and interface with the detection system. It is also essential that all eluents be degassed and they should be filtered for high resolution. The next component, important component of HPLC is the pumps actually. Now, main feature of good pumping systems in HPLC is that it is capable of output of at least say 5 into 10 raised to power 7 pascals, and ideally there must be no pulses as this may affect the detector response. So, there should be like your flow capabilities of at least like say 10 centimeter cube per minute or up to 100 centimeter cube per minute for preparative systems. Now, the pumps which are used in here are mostly constant displacement pumps and reciprocating pumps like. Now, all pumps which are used in here, they incorporate pulse dampeners to minimize the pulsing effect, and they have inbuilt safety cutout mechanism to inactivate the pump automatically if pressure within the chromatographic system changes from preset limits. Now, here is one example is that is constant volume pump. If you can see here this constant volume pump, displacement pump, it maintains a constant flow rate through the column irrespective of the changing conditions within the column, and this is which is like constant volume pump. It is a motor driven pump, we can say it is a syringe type pump that delivers a fixed amount of volume to the column by piston driven by a motor. So, what happens is this pump contains a screw jack, it contains a screw jack driven by a stepper motor. So, on the delivery stroke, what is done, it on the delivery stroke the eluent from here goes and it is delivered to the column at a constant rate. Once it is done, then these valves will be then accordingly they will be closed or opened, like say for when it has to be the eluent has to come from the reservoir, this will be opened, and then in the next when the syringe pump strokes and like when it has to deliver this displace this eluent onto the column, this valve will be open and this will be closed. So, this is like constant volume pump and it is quite used and in the HPLC systems. There is another one which is called reciprocating pump. Now, in reciprocating constant displacement pump, a motorized crank drives the piston and check valves regulate entry of eluent to the column. On the compression stroke, like here this one is a drive motor and there is a crank driver motorized crank which drives the piston. So, what happens is on compression stroke the eluent is forced from the pump chamber. So, eluent will be forced from the pump chamber to the outlet valve and to the column, and during the return stroke the exit check valve closes and the eluent is drawn in via the entry valve, that is the inlet valve. So, these two strokes, that is compression and return stroke, one will let the eluent come into the system and another will force the eluent into the column. So, these pumps produce small pulses of flow and pulse dampeners, as I said, are employed to minimize the effect. So, these are the two kinds of pumps which are utilized, constant displacement pumps which is constant volume pump and reciprocating pump. So, the pump here are important in a sense, they will maintain the constant flow rate irrespective of the conditions in the column, and there is a cutoff limit that if the pressure goes higher after certain set limit, then pumps will automatically switch off. Another important component is detectors. There are a lot of different kinds of detectors which are used. Now, the quantity of material applied to these columns is normally small, and it is very important that sensitivity of the detector is sufficiently high and stable to respond to the low concentrations of each analyte in the eluate. Now, we are taking some examples of detectors. Most commonly used detector is the variable wavelength detector. It is a UV visible detector based on UV visible spectrometry. It is capable of measuring absorbance down to 190 nanometer and can give full scale deflection for as little as 0.001 absorbance units. Sensitivity is almost like 5 into 10 raised to power 10 gram per centimeter cube, and linear range is of 10 raised to 5. So, they allow continuous monitoring of the column eluate. There could be other detectors like scanning wavelength detector, and it has facility to record the complete absorption spectrum of each analyte and display a plot on computer screen in real time. Then there are fluorescence detectors which are extremely valuable for HPLC because of their greater light sensitivity, like in order of 10 raised to minus 12 gram per centimeter cube. Then UV detectors, but they have reduced linear range, and they could be used, but limitation is that not many analytes fluoresce. So, that is the limitation. So, they are used for specific analytes. Then there could be electrochemical detectors which are selective for electroactive analytes and are potentially highly sensitive. There are two types, which is amperometric and colorimetric, and they have amperometric detectors have higher sensitivity, and then the colorimetric, and these are also have greater linear range. Then also you can attach to HPLC mass spectrometry, and HPLC like here you will enable the analyte to be detected and its structure determined by mass spectrometer. You can it's like it's a mechanism for identification of different kinds of analytes, and also you can see overlapping peaks here. Then there could be like certain times you can use NMR spectrometer directly attached to HPLC, and it can again give structural information about the analyte that is complementary to the obtained by HPLC MS, that is mass spectrometry. Then there could be refractive index detectors, and they depend on change in the refractive index of the eluate as analyte emerges from the column. It will respond to any analyte in any eluent, and the changes in refractive index being either positive or negative. Commonly used in for let us say analysis of carbohydrates and other analytes. Then there could be evaporative light scattering detectors which relies on the vaporization of the eluate, evaporation of eluent, and quantification of the analyte by light scattering could be done. It can quantify analytes in flow rates up to 5 centimeter cube per minute, very useful for the detection of say fatty acid, lipids, and carbohydrates. So, these were some of the detectors which could be used, but mainly it is a UV vis detector which are the most common, and for say carbohydrates refractive detectors could be based on refractive index change could be utilized. Then fraction collector is utilized to collect the fractions of the analyte, purified fractions. Now, for studies in which analyte in the eluate is to be collected and studied further, that could be divided into different fractions, and automatic fraction collectors which are programmed along with the whole system, and they could be designed either to collect a selected volume of eluate or to collect the eluate for a predetermined period of time. So, both ways it could be done. So, this was about the high performance liquid chromatography, and you could see that there are a lot of changes in low pressure and high performance liquid chromatography in terms of particle size, in terms of flow rates, in terms of types of pumps utilized, in terms of high sensitivity detectors utilized, and in terms of purity and the high resolution. So, there are a whole lot of differences, and HPLC is widely used for high resolution work. So, both low pressure chromatography or high performance chromatography, there are a whole lot of different chromatographic methods have been developed for both of these techniques, that is low pressure and high performance. I'll just give you certain examples here of certain chromatographic methods which are widely used, and we will be also discussing some of them in the subsequent lectures. One is adsorption chromatography. Now, adsorption chromatography is probably one of the oldest types of methods, and it utilizes a mobile liquid or gaseous phase that is adsorbed onto the surface of a stationary solid phase. The equilibration between the mobile and the stationary phase accounts for the separation of different studies. Like you can see here, there is a adsorption, there is surface on which this material or solute will adsorb onto, and this is the basis of separation. Next is partition chromatography. Now, this form of chromatography is based on a thin film. You can see here thin film formed on the surface of the solid support by a liquid stationary phase, and the solute equilibrates between the mobile phase and the stationary phase. So, you can see here solute is dissolved and in a liquid phase coated on surface, and mobile phase passes on, and this becomes basis, like for example, reversed phase chromatography and other systems. Then there is ion exchange chromatography. Here what is happening? You have immobilized the ions actually, like the use of this it is a covalently attached anions or cations on resin, and the solute ions of the opposite charge will be attracted and will be binding to the stationary phase, and then you will have to have a they will be binding through electrostatic force, and then you have to have say salt or some other eluent containing those material to elute the analyte. So, this is another important chromatography which we are going to discuss. Then molecular exclusion chromatography. It is also known as gel permeation or gel filtration chromatography, and it lacks an interaction sort of thing between a stationary phase and the solute. But what happens is here the the separation occurs through a porous gel, and the stationary phase is the solvent or the mobile phase trapped in the pores. So, pores are normally small and exclude the large solute molecules, but allows smaller molecules to enter the gel, causing them to flow through a larger volume, and this causes the larger molecules to pass through the column at a faster rate than the smaller ones, because larger molecules will not be able to enter the pores and will not be retained for a longer period of time as compared to smaller molecules. You can see there are pores, and these molecules are kind of entering and going out. So, they spend more time in the column as compared to the bigger particles which will not be entering these pores. Then there is affinity chromatography, and it utilizes the specific interaction between one kind of solute molecule and second molecule that is immobilized on a stationary phase. For example, the immobilized molecule may be an antibody to a specific protein, that is antigen, and when solute containing a mixture of proteins are passed by this through this chromatographic stationary phase, only the specific protein will bind and others will not bind to it. So, you can see like there is a ligand which is here, and this specific protein will only bind to that ligand, immobilized ligand. So, that is another form of chromatography which we are going to discuss. Now, if you see in this figure, it is summarizes sort of things where you have certain chromatographic like gel permeation, adsorption, reversed phase, partition, ion exchange, and here you could see increasing polarity like more polar ion exchange chromatography could be utilized, and for say non-polar adsorption and other chromatographic systems could be utilized. Gel filtration or permeation can be utilized for any kind of molecule, because it is lacks the particular interactions of that sort. So, this completes our section on introduction to column chromatography, where we have discussed the low pressure column chromatography or liquid chromatography and high performance liquid chromatography. We have gone through different components of both the systems, where we have discussed about columns, pumps, stationary phase, mobile phase, detectors and so on. And in the next section we are going to discuss, we will start with a specific chromatographic method, which we are going to discuss about ion exchange chromatography, gel filtration or exclusion chromatography, affinity chromatography, and also we will be discussing about gas liquid chromatography systems. Thank you.